The evolution up to modern man is not as straightforward as one first assumes. In contrast to a linear development, there were always flowing transitions that either prevailed or died out. Of course, the evolution from Australopithecus to Homo sapiens is an exciting story. But today, this video is about a colleague who lived parallel to modern humans, also called Homo sapiens, and even to the Neanderthals until 40,000 years ago, the so-called Denisovans. The first bone fragments of Denisovan people were found in a cave of the same name, Denisovan Cave, in southern Siberia, near the borders of Mongolia and Kazakhstan, in 2010. At that time, it was not sure whether that was a Homo sapiens or Neanderthal fragment. However, through genomic studies it became clear we have found something completely new here. It is known through studies that many people carry a small amount of Neanderthal DNA. Therefore, the exciting question regarding our new bone fragment is, how far have the Denisovans come and how much of their DNA do we modern humans carry? According to studies, the genomes of people from island Southeast Asia seems to have a Denisovan signal in it. The geography of island Southeast Asia is a very special one. Here this line is also called the Wallace Line. This is formed by an enormously strong Indonesian current. This line acts as a kind of a border for the Eurasian placental mammals. It's one of the world's biggest disjunctions. This line here is the Lydekkers line. This is a boundary for the marsupial mammals ecosystems. For example, a kangaroo is a marsupial. The region in between is called Wallacea. If we look at how much Denisovan DNA has been found relative to the rest of the genome in modern populations, we can see a trend. On the mainland continent of East Asia, there is virtually no Denisovan signal. However, if we look towards Wallacea and island Southeast Asia, the Denisovan signal in the genome is getting stronger. So, how does the human body and genome function as a kind of cap service for parts of already extinct human lineages? To find out how much Denisovan DNA can be found in modern populations, the scientific team around Guy Jacobs has created a complicated and complex workflow. First, high coverages genomes were collected from 14 different islands from Southeast Asia from 161 individuals. Of these 161, a significant proportion were even ethnic groups. Sequencing was performed using Illumina technology. This produced an incredible number of short but high-quality DNA reads. These were combined with 317 other previously published genomes related to human genome projects from the region. As only high-quality reads were used, they were also checked for their quality. Finally, these reads were aligned with the GRCH37 human reference genome. However, it was found that not all of the 161 original genomes were suitable. Through a kinship analysis, 5 genomes were removed from the dataset as these were first-degree relatives. This could have strongly influenced the result. This means that in the end we had 155 new genomes and 370 published genomes left, in total 471 genomes. However, the ancestors of the ICEA individuals put the project to a huge test. It was speculated not only that Denisovan genes would be found in the individuals, but also Neanderthal genes. Since Neanderthals and Denisovans are quite closely related, a Denisovan signal can be misinterpreted as a Neanderthal signal. Therefore, these two ancestors and their signals had to be separated from each other. Here we have an example with 4 different populations, one from Europe, one from Siberia, one from East Asia and one from Papua. Guy Jacobs' team pulled something incredible off. They looked for so-called introgressing blocks in the genome in which a mixture of Neanderthal or Denisovan DNA could be seen with modern human DNA. This makes it easier to assign ancestry to individuals. Based on these blocks, 3 different statistical methods were then used to filter out the Denisovan signal, if it was there. First, they used a tool called chromopainter. This tool can be used to identify haplotypes that are more similar to the Denisovan genome. Second, a hidden Markov model was used to find the same signature. Last but not least, S-star was used to identify clusters associated with non-African variations. All of those steps were done with the Denisovan and the Neanderthal genomes in comparison to our 471 genomes. The combination of all of these methods enhanced the Denisovan signal by a factor of 50. What was the result of the whole analysis? First of all, we have not only one Denisovan signal, but three. These three populations can be divided as follows: D0 as the Altai lineage East Asia, D1 less divergent from the Altai population, and D2 more divergent from the Altai population. The first split between D2 and D0 seems to be 363,000 years ago. About 80,000 years later, 283,000 years before, D1 has split off from D0. The interesting thing is when the Denisovan population mixed with modern humans. What we are seeing here is a rough timeline when and where exactly the modern human populations are splitting up. The two Denisovan lineages D2 and D1 introgressed at different times with the same modern human population. So the first mixing between D2 and the modern humans was in Papua about 46k years ago. The second mixing then occurred with the D1 population about 30k years ago. This also happened with the Papua populations. The D0 populations seem to have mixed with the East Asia populations between 20 and 10k years ago, but this is only an assumption. All in all, an interesting geographic pattern can be discerned. The D2 lineage, which introgressed earlier with the modern humans, dominate island Southeast Asia. The later lineage, D1, is much more dominant in Papua New Guinea. About 4% of the genome of the present modern population of island Southeast Asia is populated with Denisovan signals. Amazing! All these results suggest that Denisovans did not stop at geographical barriers. It remains exciting where we will find this exciting but also strange group in the future. I thank you for your attention and I hope that I could bring you a little bit closer to the exciting but also complex population genetics of the Denisovans. How much non-Homo sapiens do you think is in you?